Hello, and welcome back to Beyond Networks, the evolution of living systems. This is sort of part two of the last lecture, but also part two of the module about mechanisms as processes and dynamical systems. And here, we're going to take what we've learned from the pendulum about dynamical systems into the world of gene regulatory networks. And we're going to look at a very simple model of gene regulation and see how we can explain the features of gene regulation using dynamical systems, and in particular, the basic feature of a dynamical system, which is defined by its flow. Remember, the flow is the totality of its trajectories, and if you know the flow, if you can draw the phase portrait of a system, then you understand its dynamical repertoire. You understand what it can do under all kinds of specific circumstances. So this is what we're trying to do. And this is what dynamical systems models, a type of formal model, does for the sort of dynamic mechanistic explanation that we said in the previous module we wanted to have for developmental processes. So let's start this lecture by giving a proper introduction. Although we've briefly encountered this animal before, I've never properly introduced it. This is a vinegar fly, Drosophila melanogaster. The adult is beautiful, red-eyed, beautifully segmented body plan. You can see this by the stripy pattern on the abdomen and the arrangement of the legs and the different thoracic segments. Standing on a banana leaf, but it doesn't eat fruit. It's not a fruit fly. It eats microorganisms that grow on rotting fruit. So it's not a fruit parasite like proper fruit flies, tephritid flies. It's a detritus eater, basically, living on rotting fruit. And that lifestyle has some implications. First of all, fruit flies need to develop extremely quickly. So the whole of embryogenesis, from egg laying to hatching, happens in 24 hours. The whole developmental cycle, life cycle, it's only about 2 weeks long. And so the basic body plan is laid down during the very early stages of these 24 hours in the blastoderm embryo of the fly. So here you see the early cleavage stages, the first 3 hours of Drosophila development. These are Drosophila embryos, the head end at the top, the tail end at the bottom. What's unusual here, two things to note that are unusual. One is this entire embryo that's almost 0.5 millimeter long, it's visible by the naked eye, is one single cell. Okay, so what you have is an incredibly fast succession of nuclear divisions. The nuclei are visible here. They're fluorescently labeled in the early cleavage stages. But no membranes exist between the nuclei, so this embryo is a syncytium, a multinucleate cell. These divisions are extremely fast. They happen every 12 minutes. As far as I know, this is one of the fastest divisions in the animal kingdom, or in the domain of life in general, I think. So these nuclei copy their genome and they continue on to the next round immediately. Incredibly fast. Also, if you remove the DNA from these nuclei, the whole structure that you see here, first, this sort of arrangement of nuclei, the spindles, and then the migration of the spindles and the nuclei to the periphery happens without any active DNA in the embryo. So it's sort of programmed into the embryo by the mother. This is an amazing feat of self-organization. So this is the sort of challenge we have in biology. We want to understand how this happens. What is driving this? Okay, not a genetic program. You can remove the genes. It's all self-organizing. It's an absolutely mind-boggling, amazingly complex self-organizing process. Okay, so by this time, by cleavage cycle 9 and 10, the nuclei have arrived at the periphery of the embryo, and they form this layer of nuclei here at the periphery, which is called the blastoderm. So this stage here is called the syncytial blastoderm stage. Most of the nuclei are at the periphery. There's some that are left back here in the yolk. They're very large. You can see them faintly here. This is basically food for the embryo, but the embryo proper is localized on this surface structure called the blastoderm. And what's also happening during these 1.5 hours, is that a genetic cascade patterns the segmented, lays down the segmented body plan of the animal in 1.5 hours. This is an incredibly fast patterning process, extremely fascinating. The second advantage of this system is not only doesn't it have membranes between the nuclei, it also doesn't grow, it doesn't have any sort of major tissue rearrangements if you only look at this stage and not at the earlier migration of the nuclei. Very simple, as simple as it gets in the animal kingdom, very fast. Okay, so by the end of this stage, cell membranes form, cellularization occurs, and gastrulation starts happening towards the end of cycle 14A. So this cycle continues after gastrulation. And basically, what you see during this red phase here is the appearance of the first proteins from genes that are zygotically expressed before everything is maternally preloaded, okay? So an extremely unusual but very fascinating process. And also very famous, of course, because of the pioneering and Nobel Prize winning work by Nusslein, Volhard and Wieschaus, Janine Nusslein-Volhard and Eric Wieschaus in the late 70s performed a famous saturation mutagenesis screen where they saturated the genome of Drosophila with mutations hitting every single gene at least once and then scoring, looking out for segmentation phenotypes. So they identified a complete list of necessary genetic factors that are involved in this process and classified them according to the different phenotypes they produced. For example, when entire parts of the embryo are missing, the genes were classified as gap genes. When every other segment was missing in the embryo, which is an interesting phenotype, they were classified as parallel genes. And if they affected the polarity of every single body segment, they were classified as segment polarity genes. And in this way, by first classifying the phenotypes and then studying mutants of every one of these genes and how they affected the expression of other genes, Drosophila geneticists worked out a parts list, a very detailed parts list of genes and their interactions, a gene regulatory network that you can represent in a graph, like we've seen before. But what we didn't know until about 20 years ago, when I started my PhD, is how we could put that system together again and check whether the predicted sort of interactions actually produced the segmental pattern that we see. So here's the system from another point of view. You see its regulatory cascade. There are 4 layers. Maternal coordinate genes express proteins that are present in the embryo in these broad gradients. Bicoid is shown here. So the RNA is localized at the interior of the embryo. It starts to be translated into protein, and the protein, a transcription factor, is then diffusing through the embryo, forming this beautiful nuclear gradient across the entire embryo. And you can see the dots here, the nuclei with different concentrations of bicoid. There's a complementary gradient of caudal from the posterior and a second system called the posterior system. So bicoid, caudal are the anterior system, hunchback from the posterior system has another gradient from the anterior. Then these transcription factors of the maternal, encoded by maternal coordinate genes, these maternal gradients activate the gap genes that are expressed in these sort of broad overlapping domains. These are hunchback, Krupel, Knirps, giant, tailless, and Hookabine. And these gap genes not only regulate together with the maternal genes, the parallel genes, but also themselves. So there's regulation not just between those layers of this regulatory hierarchy, but also within the layers themselves. So these two maternal coordinate genes and gap genes together activate parallel genes in this periodic pattern of 7 stripes. There are different gene names here: even skipped, odd skipped, hairy, runt, Fuji, Terazu, and paired, which expressing these beautifully also staggered 7-stripe domains. And all of these genes together then lead to the expression of the segment polarity genes, in particular engrailed and wingless, which complement each other and form this beautiful 14-stripe pattern here. At this point, gastrulation has occurred, the embryo has wrapped around the eggshell, posterior is now here, and these 14 stripes define the formation of the parasegmental boundaries and ultimately segmental boundaries later in development. So this is called the segment determination system, the segmentation gene network of Drosophila melanogaster, a textbook example of a very well-studied genetic regulatory network. And what we're going to do is we're going to focus in on the top layer, sort of this transition from these maternal gradients onto these overlapping, broad overlapping gap domains here. So, as I said, genetically, this whole sort of interaction network was worked out. So what is the problem? Why would you go and model this system? Okay, and so what I'm going to talk about is work that started when I did my PhD. I started in the year 2000 with this work in the lab of John Reinitz at Stony Brook University at the time, near New York on Long Island. And what we did is we tried to reverse engineer the system in a sort of an unusual way. We didn't do genetics. We tried to reverse engineer the system by modeling, by creating models that we fit to data that would reproduce the patterns that we saw. And what we're going to do right now is we're going to take a subset of these genes and quickly reverse engineer the gene network underlying that system, producing this pattern. So I'm going to teach you the principle of reverse engineering here without any math. Okay, and what you do is you start with a wild type embryo, a healthy embryo, and you look at the expression pattern of the genes. Okay, so what we have here is a beautiful image of an embryo. The head is here, tail is here. You can see one cell, multinucleate, many thousand nuclei, and each nucleus is visible as a dot. And this embryo was immunofluorescently stained with antibody against two proteins that are encoded by the gap genes. One is Krupel, which is colored green here in the middle, and the other one is called Giant, which is expressed in this beautiful stripy pattern, especially here in the head. And so what we want to do is these are two transcription factors. What I can tell you is they regulate each other. We want to look at this picture and infer from that what the network looks like that connects those two genes. So think about it for a second. What is the most striking sort of aspect of the expression patterns of those two genes? Especially in the middle of the embryo, you see that there is not a single nucleus that expresses both of them at the same time. So either you are green or you are blue, even here in this intermediate region, where both of the genes are weakly expressed, there is not a single nucleus that has both of them present at the same time in any detectable way. So what must be happening here, and this is why I chose this system, it's quite simple to infer the network. What must be happening here is that these two transcription factors really strongly inhibit each other. So here I've drawn it up. One transcription factor one gene produces the blue factor, and the other one produces the green factor. In this case, green Krupel protein dominates, binds the giant, switches it off, and you have nothing but green protein present. Okay, no expression here. For giant, a lot of expression by Krupel, or the other way around. So basically, the network looks like this. You have two factors, and they repress each other very strongly. And you put this in a spatial context, and you get this beautiful complementary pattern here. Of course, the situation is more complicated out here in the head. We're not going to talk about that. We cannot explain these very beautiful stripes here, just using those two genes. But just look at the middle of the embryo. Okay, so this sort of network, this mini network, it's part of the AC/DC circuit, remember? It's a double negative feedback loop. We were talking about this. Double negative means a positive feedback loop. That means it acts as a switch. Either green or blue is present, but never both. So positive feedback here, switch-like behavior, and this is why this beautiful little circuit is called a genetic toggle switch. And we can do a lot of work with this. So we're going to spend the rest of this lecture examining the behavior of this very, very, very simple two-gene network, even simpler than the AC/DC circuit, to which we're going to come back later. Okay, so how can we study gene networks? Again, at the end of last lecture, I told you a little bit, there's very different sort of strategies to do this. We could just model them using differential equation, assuming that there is no noise in the system. We're just interested in the sort of average behavior of the system, right? We can make a very detailed biochemical model based on the chemical master equation. That is a stochastic differential equation that takes into account a realistic model of the molecular events that are going on underneath. But in this case, unfortunately, we don't know enough about the precise biochemistry of eukaryotic transcriptional regulation that we can do this. Or we can take an intermediate approach and do stochastic simulations using the Gillespie algorithm, for example. So different ways of modeling this system, okay? We're gonna ignore noise completely at the moment, because we're gonna keep the system as simple as possible. And also I would argue, if you do not understand the sort of average behavior, in most cases, you shouldn't worry about the noise yet. I mean, this can be dangerous, okay? We have to be careful with that, because in some cases, the noise is not noise. Noise is a negative term. The fluctuations that occur in the system are driving the pattern forming capabilities of the system. So we cannot exclude this. But in this case, I can tell you they're not the main thing. So it is possible to understand the system without considering the fluctuations in the system. So we're going to focus on ordinary differential equation models. Okay, so we have this little toggle switch model. It's a little bit more complicated than what I showed you before, because we allow auto-regulation, auto-activation to occur. Also, these genes, of course, have to be activated in the first place. So there is some sort of term here represented by these alphas that are constitutive activation terms. And then, of course, the proteins that are produced by these two genes are degraded with a certain decay rate. Protein decay is something that people always forget, especially geneticists never consider it, but you have a patterning system here that makes a very intricate pattern in 1.5 hours. So these patterns, they change on the order of 2 or 3 minutes. So these proteins have to be extremely unstable if you want to pattern this fast. If you have proteins that are too stable, the pattern can't change at this time scale. Okay, so these are things you need to consider, that factors that go beyond the normal sort of genetic factors that you would think about. Okay, so you can formulate this as a set of differential equations, and we've encountered differential equations with the pendulum. Now we're going to use a different notation. Okay, so we used a sort of a physical notation before with the dots. Now we're going to write it, the change of concentration, protein concentration x over time. We're going to write it like this. dx over dt means this equation describes the change of the concentration of x over time t. The same here, change of the concentration of y over time t. There is factor x, factor y. The protein for Kruppel, the protein for giant, they both have an equation, and they have an equation in each one of the nuclei in the embryo. Okay, and their expression and regulation here is represented by these sort of complicated equations that we're going to take apart quickly now. So the basic ingredient of any dynamical system, I told you, are its state variables. These were position and velocity for the pendulum. Here, they are the concentration of the two factors involved in the regulatory network, the concentration of x and the concentration of y. These are called the state variables. They are what is changing. They describe the changing succession of states over time. Then we have parameters of the system. All these letters here describe, for example, the strengths and the nature of different regulatory interaction, A, B, C, and D. They are regulatory parameters, but also the decay rates, for example, of these proteins. They're very high because, remember, the proteins have to be very unstable. These are called the parameters of the system. In the case of the pendulum, that was the weight of the bob and the length of the rod. Remember? Parameters. Parameters usually don't change over the time of a simulation, but we'll come to an exception next time. And then we have what are called boundary conditions, sort of influences from outside the system onto the system. And in this case, we have these constitutive activating terms that represent maybe the activating input of maternal gradients that we do not represent explicitly in the model itself. So we've decided to exclude them from the model, although we know they're important, and we just represent them as these sort of boundary conditions, a magical sort of influence onto the formal model from outside its boundaries. Okay? Parameters and boundary conditions together are often counted as parameters of the model. Okay? But boundary conditions are parameters that are a little special, that specifically describe influences from outside the system or influences from the shape of the domain on which the system occurs. Okay, so what do these equations describe? Okay, So the left-hand side of the equation, as I said before, represents change of the state variables over time. So change in x, change in y over t. Okay, so there is one equation for the change of each state variable. That is how you formally describe a dynamical system. And the right-hand side of the equation represents all the factors, all the influences that change this change depends on. Okay, so if we dissect it, there's first of all, there's a repression. Okay, so these two factors repress each other, and the particular mathematical term that you see here codifies represents a sigmoid repression curve. So if x is present at a certain above a certain threshold, the production of y or f of x here will be shut off, and the other way around. Okay, so this particular term here is called a Hill function, and it represents a sort of a sigmoid leaky switch-like behavior of the two genes. Hill functions are nonlinear functions. They are what makes this equation, this system, a nonlinear dynamical system. Okay, so it will exhibit threshold and unexpected behavior compared to a linear system that you can always predict. There's a second term for activation, also a Hill function. I'm not depicting it here, but it's just sort of flipped around. If you mirror these two graphs, the same thing. So you have activation terms you see. In this case, it's auto-activation. So Y, C is the term that represents the auto-activation of Y. Okay? You have protein decay in a separate term, minus, because the protein is getting out, is disappearing. And the constitutive activation terms were also represented in this equation. So basically, we now have a formal mathematical model that describes this toggle switch, and we can simulate it, okay? And we can start from all kinds of different initial conditions. And we want to find out, what does it do? Remember from the pendulum, what do we need to do to find out what it does? We need to draw its phase portrait. And just like the pendulum, we have two state variables here, which is very convenient because we can draw the phase portrait of the system in the phase plane. The phase space here is a phase plane. It's defined by the concentration of factor y, which is crucial in green, and x, which is giant in blue. And then you can simulate the system, and depending on where it goes, you can draw these different arrows onto the phase portrait, and you will always end up at these two blue points. So let's sort of look at this phase portrait in a bit more detail. Of course, the arrows represent the flow of the system. These circles and eye-like and oscillatory trajectories in the case of the pendulum. Same thing here. And these trajectories, the flow, they all go to the middle of this phase plane very quickly here, and then very slowly out to this blue point. So each individual path here is a trajectory of the system. Depending on where you start, if you start with a little bit more blue factor than green, you always end up with this blue dot. When you start with a little bit more green, you end up with this blue dot. And that represents the toggle switch behavior of the system. It is due to there being two attractors present in the system. One has a lot of giant and no crucial, and one has a lot of crucial and no giant. So they're mutually exclusive. All of the trajectories, almost all of the trajectories end up at one of these two. And the only thing that determines where you end up at, is whether you started with a little bit more of the blue or the green factor. Okay, if you have more blue than green in the beginning, no matter how much of the protein you have, you'll always end up right here. This explains the robustness of the system. No matter where you start, you will go to this one target point. Okay, it's very robust, it's very resilient. There's a special case in the middle. So these are fixed points of the systems, and they're called attractors, because if you push the system away from them, they return. Remember the centers of the pendulum, they were not attractors, okay? Because if you push the system away, if you do push the system, a linear pendulum without friction will infinitely keep oscillating around that center. It will never go back. But here, if you push the system off the attractor, it'll return to the attractor. There's a third fixed point in the middle. It's called a saddle point, and I'll show you why in a minute. that is very special, okay? So you only end up there. Remember, this system has no fluctuations. If you start exactly with the same concentrations of the green and the blue factor, exactly the same amount of purple, same amount of giant. You go along this line and you end up here. But if you give the system the slightest push from there, it doesn't go back to the saddle, but it converges immediately to one of the attractors. So this is an unstable fixed point. Just like in the pendulum, the equivalent was this fixed point on top. That was a saddle point too, remember? If you push it off, the system will diverge very far away from the original position. Same here. Okay, so fixed points are extremely important. We said with the pendulum that if you understand the fixed points and sort of how you get to the fixed points of the system, then you know what the system does. There are different types of fixed point. We've encountered a few of them already. So what we have here are what are called classical fixed point attractors. There's a bunch of trajectories, they come in to this one attractor. You can have different sort of shapes of that. So other points. So this one is very peculiar. It's a star. It's very unusual. It occurs very rarely, so you won't probably encounter it anywhere. So all the trajectories go straight into a star. Very unusual structure. You can have a non-localized fixed point, which could be a line, okay? So no matter where you are on the line, if you get pushed off in this way, you return, but if you get pushed off a little bit diagonally, you return to a different point on the line. You could have a center, just like in the pendulum before. So the centers are typical for conserved energy systems. They occur in physics, but basically never in biology. So don't worry too much about them, okay? You could have a spiral sink, which is very interesting. So it's like a fixed point attractor, but the trajectories around it spiral into it. Interestingly, they only reach the point itself at infinite time in such an idealized system. Okay, so they never really reach it, but they spiral and spiral and spiral and spiral. You zoom in, it'll always look like this. Okay, very fascinating sort of attractors. And you could have a saddle point, unstable fixed points where you diverge if you get pushed off in a certain direction. Okay. There are higher dimensional attractors as well. So we're going to focus for the rest of this lecture only on these sort of fixed points. So don't worry about the other ones. And the star is extremely rare, okay? So if you go to higher dimensions, you have other interesting attractors. So in 2 dimensions, you can have circular trajectories that are attractors. So basically, this is called a limit cycle. Basically, the system stays on the cycle. If you push it off, it'll return to the cycle no matter where you are. And this is a sign of a stable oscillation of the system, limit cycle oscillation. We'll find that, we'll encounter that later on. You can have 3-dimensional structures. This is a limit, not a limit cycle, but a limit torus. So the system is on this torus, wherever it is, if you push it off, it'll return to it. And most beautifully, in 3 or higher dimensions, you can have what are called strange attractors, which are attractors that have a very sort of weird structure. They're confined to a certain area of phase space or state space. But if you start on a trajectory within the attractor, it'll never repeat, okay? So it's infinitely complicated. And these are the sign of chaos, deterministic chaos in dynamical systems. We won't, unfortunately, talk about this very fascinating topic a lot, but if there are celebrities among the attractors, possibly this one. This is the Lorenz attractor. It was one of the first strange attractors to be discovered by Ed Lorenz, and he found it in a very simple model for a meteorological system. And it became, because it looks a bit like a butterfly, it became this sort of celebrity attractor because of the butterfly effect. It represents the instability, the chaotic nature of the weather, so that if a butterfly flaps its wings in the Amazon, you could get a hurricane a few months later in the Caribbean. So this sort of cascading effect, unpredictability in chaotic systems became sort of emblematic. It's one way in which we cannot predict the future, even if we don't even consider noise in dynamical systems. Deterministic systems can be unpredictable, which was an amazingly fascinating insight at the time. The thing is, biological systems, as far as we know, there's not much chaos happening because biological systems don't like chaos. or unpredictability, of course. They fight unpredictability. And we'll see several ways in which organisms can do this when we talk about organisms as dynamical systems. But coming back to our really simple toggle switch, it doesn't have any chaotic attractors or anything like that. We just have two fixed point attractors and a saddle. And as I said, the behavior of the system is very simple because there's only two basins of attraction. A basin of attraction consists of all the trajectories that end up at a specific attractor. And in this case, everything that starts with a bit more blue than green ends up here. Everything that starts with a bit more green than blue ends up here. So you have two basins of attraction, and the line that crosses through the saddle in the middle here is called the separatrix. Okay, a bit more vocabulary. You have these very fascinating valleys here in the middle. So if you get pushed off the saddle point in a straight way, you go straight into the attractor here or here. And these special trajectories where the flow is very, very slow, you will notice, they're called unstable manifolds, and they will become important in just a minute. Okay, so we have this fascinating sort of structure, the phase portrait. And I said attractors are extremely important, but there are two problems with attractors in biological systems. First, we do not know that these systems are actually at fixed points when we look at them. So we cannot assume that they are at steady state, okay? So it's very important to also consider the transient dynamics of how systems get to an attractor, okay? So in this case, if you look at our system, it's a switch, a toggle switch. So if we start here very close to this separatrix, we move very quickly into the valley, this unstable manifold, and then very slowly out here, the time is represented by color here, and you can see the trajectories of x and y. So for quite a while, these factors can coexist and only separate out after a while. So you have to consider this. Another reason why you cannot assume that this toggle switch will automatically lead to toggle switch-like behavior. If you see this structure in a network, it could be that the developmental system never reaches the steady state and sort of stays in this area here where both x and y are present, which is something you would not expect to happen from the network structure alone. But if you add a little bit of delay, so let's say the effect of x on y has a little time delay, then crazy things start to happen, okay? You can get oscillations of these factors before they settle down at their fixed point attractors. Normally, you only get oscillations in systems that have limit cycles or spirals, okay? Pseudo oscillations and spirals, but here you can get transient oscillations, damped oscillations before the system settles down with fixed points. So again, this is a positive feedback loop, double negative. It's supposed to switch, not oscillate. This system that is supposed to behave as a well-behaved switch can oscillate transiently before it settles out to its steady state. Always expect the unexpected. Structure does not determine function. Repeat after me. Structure does not determine function, especially if you consider the transient behavior of the system. And in biology, you can never assume the system is at steady state. Okay, so let's wrap up here. I've taken you for a wild ride into the world of dynamical systems theory and mathematics. I've introduced all these terms: separatrices, saddle points, attractors, basins of attraction. What the hell? So let's connect it back to biology. And for that, we need to introduce one more sort of way of depicting the phase portrait of a system. Practically only possible for systems with two variables, not more. That's why we chose such a simple system. So in this case, you can interpret the flow of the system, these arrows, as slopes on a landscape. And if you draw the system like that, so you're basically drawing it in 3 dimensions now, with the arrows representing slopes, it looks like this. This is called a potential landscape, and because of the way it was calculated, it's called a quasi-potential landscape because it's not properly derived, but don't worry about that. So a potential landscape of this phase portrait looks like this, and you see now why the saddle point was called a saddle point here. You have very steep slopes here that go into these flat valleys that end up the deepest points here at the attractors of the system. And you can see now how this toggle switch works. You roll down here and then slowly out here. That's how the ball rolls down the landscape. So what we've done here for a very, very simple system is we've reconstructed a potential landscape. And this potential landscape, of course, is nothing but a precise mathematical formulation of Waddington's epigenetic landscape. The details look different, okay? And also, very importantly, this is no longer a metaphor, okay? It represents something that we've measured out, we've observed from the behavior of the system. Just like with the pendulum, we don't need any maths. We can infer this landscape from observing the behavior of the system, the time development of the concentrations of these two different factors, independent of the mathematical formulation. We can reconstruct this potential landscape. So these attractors, these basins of attraction, these potential landscapes, they are real in the same sense, or even more so than genes are real, especially more real than enhancer elements. We don't know what enhancer elements really are. There's no good definitions. I can measure these features of phase space from observable expression patterns. So in the sense of Wimsatt, I can derive them in many different ways. Mathematically, derive them, I can observe them, I can derive the landscape from there. Many different ways to do this. Okay, so this is a robust and therefore real feature of the system. Attractors are real, okay? They're not some sort of abstract notion. The way we formalized it here is an idealization and an abstraction. But these attractors exist and they determine what the system can do. And if we can measure them, if we can infer them somehow robustly, we understand what the system does. And this is how these dynamical systems models help us explain mechanistically the processes of development. They are not themselves mechanistic explanations. We'll come back to that in the next lecture. And of course, once we have this landscape, we can also check how it behaves when we start changing the strength of the interaction. So what I'm going to show you here is a simulation where these autoregulatory terms on the two factors increase over time. Or maybe I'm not actually right now. I will show you in a later lecture. So you can imagine that when the parameters of the system change, you get a change in the landscape, just like you would explain this sort of thing in Waddington's metaphor of the epigenetic landscape. So I will show you that movie in a later lecture when we talk about time dependence of systems. Actually, the next lecture. But let's wrap up for now because I've been going on for quite a while and it's been quite dense over the last two lectures. So where are we at right now? So I told you that trajectories form the flow of a dynamical system, and the dynamical system is its own flow, right? So attractors determine the outcome of the flow, but be careful. The system could behave in a transient way. It may not be at those attractors. It may never have the time to reach those attractors. The arrangement of attractors and their basins with respect to each other, that's a new term. We call that the topology of the system. Okay, does that ring a bell? So the topology is the general shape of that, the general geometry, just in terms of the basins of attraction and how they stand with respect to each other. This topology determines the types of behavior the system can exhibit. And these different types of behavior are the system's dynamic repertoire. You can now take these topologies and classify different systems into according to what they can do. And of course, this is again a type of topological explanation. You've abstracted away, far away from the causal interactions underneath. And you're using these very abstract topological properties of the system no longer on a network graph, but defined in this abstract space, which is called phase space. You use that topology to classify system and make conclusions about what they can do. It's exactly equivalent to topological explanations in the network case. So by itself, this doesn't give you a mechanistic explanation, but by using these models and connecting them back to the causal structure of the network, which is something we will do only in the next module. By doing that, they contribute to dynamic mechanistic explanation. Before we can get back to that, we need to introduce two more complications. One is sort of transience, time dependence of system, which makes the parameters change of the system. So what we'll do the next two lectures is expand our horizon into configuration space. And we will look at how systems behave that change over time and what that means for us to understand sort of the principles, the rules behind the generative processes that determine developmental outcomes. Thanks for listening. I hope you tune in again next time. Bye now.